Okay, so last time we talked about horizontal gene transfer, right? So the main ways that we talked about for horizontal gene transfer is transformation, conjugation, and phage transduction. We also talked about recombination and mutations, and then how the cells repair the mutations, and then transposon. So all of these were kind of ways that the chromosome can evolve in ways that DNA can change within the cells. So, review from last time. Can anybody tell me what the first one is? Sorry, what? Yep. Transduction. So these are, just make sure you know, there a lot of them start with T's, right? So make sure you can know which one's which and tell them apart. How about the second question? C? So can you tell me what's the key to the question? Yep. So this here is a stop codon. So this is the most common one. So, knowing AUG and UAA, that's pretty much it. All right, so hopefully you all know what these other ones are, right? So you'll be expected to know what those are. Okay, so today we're going to move on into molecular regulation, and it's kind of a big area, so we're splitting it into today and next time. So today we'll start with just kind of general ideas about how genes can be regulated, and then we'll go into two examples, the lac operon and the tryptophan operon. All right, so the main idea behind gene expression and kind of monitoring when genes are made is that the bacteria essentially doesn't want to waste energy making proteins that it doesn't need at that certain time. So there are going to be genes that they kind of need for general growth, and so those things are going to be on all the time. And so when something's on all the time, that's called constitutive. And so there are certain essential genes that they need that are on all the time, but in other cases, they only need them, like, so for example, for like the flagella, right? They don't need them all the time, and so they will only turn them on when they need them. And so there are various different ways that they can actually control gene expression, and at different levels within the central dogma, right? So at the DNA level, they can actually alter DNA sequences. And so this can, again, change to either activate or disable a certain part of a gene. So they can control transcription, and so these are often regulated by either repressors or activators, and then also at the sigma factor level itself, because remember, the sigma factors are recognizing the specific promoter sequences. And then I also wanted to emphasize, just because I spent a lot of time studying this, but RNAs play a huge role in some of these transcriptional control mechanisms too. So the other ways to control the mRNA stability, right? So if you imagine that your transcript's getting made, if it degrades really fast, then there's very little time for the ribosome to translate it into a protein. And so there are different ways. It usually has to do with RNA structure and whether or not it gets degraded. But there are ways that the cell can make sure that the transcript stays around a little bit longer. Then we have at the translational level, again, so these can be either repressors or activators, but in general, what happens at the translational level, it's whether or not the ribosome binding site is accessible to the ribosome. So whether that's controlling whether that's blocked or open, and there are different mechanisms to do that. And then finally, at the post-translational level, so even if you've already made the protein, sometimes it needs extra processing for it to be activated, right? Or one thing that we'll talk a lot about in the upcoming topics is phosphorylation. So sometimes it needs to have that be phosphorylated for it to be active. Okay, so we'll start off in the beginning with altering the DNA sequence. And so one way that bacteria can alter its actual DNA sequence is through gene inversion. So like the name sounds, it actually flips the orientation of the gene within the DNA itself. And so an example of this is found in Salmonella enterica. So this is in flagella synthesis. So there are enzymes called recombinases. So this is the class of the enzyme. For this species, it's called HIN. So this actually flips the orientation of the DNA between two inverted repeat sites. So they don't do it just randomly. There's going to be some kind of sequence that it recognizes that are going to be flanking whatever gene that it's going to flip. So in this case, there are these HIX sequences that it's recognizing here. So these are going to recognize this, and I cut out all the specifics on how it actually does it. But in the end, it's just going to recognize these sequences and then flip the DNA orientation here. So you see here the promoter's up here, and now the promoter's here. So essentially now the promoter is pointing in the wrong direction, and it's probably lost its context, and so now the sigma factor can't recognize it, and you don't get transcription. Yes?
Does it flip it back?
Yep, some of them can. They will also flip them back when they need it. All right, so that was the DNA level. So now we're going to go to the transcriptional level. So the idea, and we'll just talk about some of the general terms involved in transcriptional control. But the general idea is that the cells they have to know when to turn on the genes and when to turn them off. And so they're always responding to something that's happening in their environment. So at this level, so there are two ways, right? So what's actually already inside the cell, and then what's outside in the environment. So the next couple of things we'll be talking about what's the inside the cell. So once the whatever molecules are in the cell, and this is what it's responding to and how that responds. So the main thing is that we have these regulatory proteins. So these helps the cell sense internal changes and then alter its gene expression to match whatever it needs to do. So they're usually involved, so here's the example of a regulatory protein. It usually involves a binding of a low molecular weight called a ligand. So this picture doesn't draw the actual ligand, but it kind of references it here. We'll get to the ligands in the next couple of slides. But so there are different regulatory proteins bind different ligands, and so they have to, there's specific binding that they have to find and match each other. And then, so genes encoding the proteins are usually, but not always, transcribed separately from the target gene. So this is what that means. So essentially here, so you have the gene that's encoding for the regulatory protein. This is here coming off of this promoter here. But what it's actually regulating is the gene that's coming off of this promoter here. So it's not under the control of the same promoter.
Where's the ligand?
Yes, so this one's not drawing the ligand because there's lots of, and we'll get into that in the next two slides. All right, so we'll first talk about repressors. So repressors, essentially, they bind to the regulatory sequences and they prevent transcription of target genes. So they essentially are blocking the promoter sequence, and so the sigma factor can't find the actual promoter sequence. So there are two types of repressors. So this is where the ligands come in, and it's the two different scenarios depend on how the ligand interacts with the repressor molecule. So the first one, the repressor will bind the DNA and it'll block transcription. So you got your repressor protein made from somewhere else in the chromosome. That's coming in here. If that's made, it'll come here and it's gonna bind this region here and it's gonna block transcription. In order for you to turn the transcription back on, this one requires an inducer molecule. So the ligand is called an inducer. So here, so this green molecule here is the inducer. So when this binds to these molecules, that causes a conformational change, and so it can no longer stick and bind to the DNA, and so it falls off. And so now your promoter region will be free and you can get transcription. Okay, so this is in contrast to scenario 2. So now, in order for the repressor to actually bind to the sequence, it requires first binding to the ligand. So in this case, the repressor by itself can't bind the sequence on the DNA. So it requires the ligand, in this case is called the co-repressor. So it requires this molecule here to bind here. Once this binds, then it can latch onto the DNA. And so now, in order to turn the genes back on, when the concentration of the co-repressor decreases enough, it will eventually dissociate from the repressor protein, and then this will fall off. And so this is known as de-repression. Okay, so those are repressors. These are activators. So the activators, like the name suggests, they are activating transcription, and so they are doing so. They also will bind the DNA sequence, but when they do bind the DNA sequence, they are actually interacting with the RNA polymerase and help enhancing transcription that way. So sometimes RNA polymerase has a really hard time getting started, and so these activators will come in, and whether they're like bumping into them and causing some kind of shift, all these things can happen, and it'll help the transcription get started. So in general, most activators bind poorly to the DNA unless the inducer is present. So again, in the case of the activator, the ligand is called the inducer. And so then this one again will bind to the activator, and then that complex binds to the DNA and induces transcription. And so if you wanted to stop transcription, you would remove the inducer or wait for it to degrade by that. Yes?
So what was the difference between the activator and then the first one that we talked about?
So in this case, they're binding the DNA, and then they're interacting with RNA polymerase to actually get it started. And the other ones, they're binding the DNA, and it's like a roadblock for the RNA polymerase. It's just, you can't go. All right, so that was sensing how the cells sense their internal environment with these different regulatory proteins. But the cells also have to sense what's going on outside, right? So the most common way that they do that are through these two-component signal transduction systems. So these are phosphorylation relay systems. So what happens is you have a protein that spans the membrane, which is called the sensor kinase. And so the kinase domain will be on the outside, and it will bind some environmental signal that will cause some kind of conformational change in the protein and essentially kick off this phosphorylation cascade. So you get the phosphate groups transferring from different proteins to, in this case, it's just one protein, this response regulator protein, and that's the one that can then go ahead and it can also bind to genes and either activate transcription and things that way too. So this is a huge class of proteins. There's lots of different ones that recognize different molecules. Okay, so we're gonna talk about two main examples today. We're gonna go through it carefully, and if you don't understand, please ask questions. So the first one we're going to go through is the lactose operon. So the lactose operon controls the genes for lactose catabolism. So lactose is a disaccharide of glucose and galactose. So it can be used as a carbon and energy source. So in order for the cells to use it, it first needs to let them in. So LAC-Y is this protein here, the membrane protein that lets in lactose. And then once it's in the cell has no real use for lactose itself. It needs it to break it down. And so it has this enzyme called beta-galactosidase, which is encoded by LAC-Z. And so that can cleave lactose to get to glucose and galactose. And so now the cells can use the glucose and it's ready to go. Okay, so this is what the operon looks like. So we have 3 genes here, so they're all co-transcribed from a single promoter. So here's the promoter here. The one that we care about is the promoter here. So LAC-Z, LAC-Y, and LAC-A. So LAC-A makes this enzyme, but it's not really clear what it's for. The molecule that regulates the transcription from here is the LAC-I repressor. So the LAC-I repressor is found upstream of the LAC operon. So the repressor can bind to the different, so what we call operating sites. So this LAC-O and LAC-O1, so these 2 spots here. So that's where the repressor is going to bind. Yes?
Isn't it regulates LAC-I?
LAC-I is the repressor, the repressor protein, and that's what's controlling and regulating the expression from this. Okay. So in the absence of lactose, so the LAC operon is transcribed at extremely low levels. So, okay, fine. A lot of these things, they're not perfect. So even though it's supposed to be repressing and you think that the gene's supposed to be off, sometimes, you know, it's not going to be 100% off. And depending on the use, the cells will have evolved for it to be that way. In this case, there is a reason why, and we'll get to that in a couple of slides. But how that's happening is essentially that a tetramer, so 4 of the LAC-I repressors have to come together, and so that will bind the 2 LAC-O sites, and it actually causes the DNA to make this loop, and that prevents RNA polymerase from accessing the promoter site. Okay, so in the presence of lactose, now the expression from this gene is 100-fold higher. And so what happens is that basal level of the LAC expression allows LAC-Y to be expressed. So, right, if there was no LAC-Y at all, then the lactose would never make it into the cell, right? So it makes, you know, a couple of them, has them out there just in case it needs it. And so LAC-Y will be out there and ready to let lactose in if it needs to. The other factors, so at a low concentration of LAC-Z, so LAC-Z doesn't actually completely cleave it to glucose and galactose. It essentially just rearranges lactose to form allolactose. And so allolactose now binds to the repressor. So allolactose here is our inducer. So in this case, so this is our scenario 1 of the repressors that we're talking about. So this is the inducer. So this will bind to LAC-I, and then it'll release it from the operating sites, and then you can get transcription from the rest of the genes. So there are a few other complications with this. Well, not necessarily complications, but there are a few other factors that play into the LAC operon regulation. So one is cyclic AMP. So this, in a completely separate mechanism, right, accumulates when a cell is starved for carbon. And so its name is exactly what it sounds like. So it's a derivative of AMP, but it's the 5' phosphate linked to the 3' hydroxyl, so it's cyclic. So the CAMP binds to this dimeric CAMP receptor protein. And so this complex will bind to specific DNA sequences located near many bacterial genes. So it doesn't only do this for the LAC operon, it'll do this for other sequences as well. But essentially this thing will bind here and it'll act as an activator. So, you know, we made it sound like RNA polymerase, it goes and it finds transcription and it's easy, but in reality it does struggle a little bit, and so that's why we have these activators that can help. And then we have things like for the CAMP CRP. So this will, again, it binds to the DNA and it interacts with the RNA polymerase and it kind of helps it get going and helps it form the open complex. So, okay, so this is there, but just remember, this will not be able to bind if the LAC-I is bound. So this can only help, so this can help boost, but it can't do it if LAC-I is there and blocking the promoter. Okay, and the other thing that influences the lac operon is actual glucose concentrations. So when the cell has glucose present, it's going to want to just use glucose. That's the favored carbon source for the cell, right? So because essentially enzymes for glycolysis are always being produced, so it doesn't want to waste its energy trying to catabolize lactose if it's already got lots of glucose. It's a waste of time and energy for the cell. So glucose indirectly prevents induction of the genes by keeping lactose out of the cell. So this will go back to the PTS system that we talked about before. And so if you remember the PTS system, that's how glucose comes in. And it's one of those things that transfers the phosphate bonds, right? So when glucose is present, the phosphate bonds are being transferred to glucose, right? So these enzymes here will often not actually be phosphorylated. And so the unphosphorylated form of this 2A component actually inhibits LAC-Y. So that actually prevents lactose from getting in the cell. Even if it's out there, the cell doesn't need it at that point. But when the cell's out of glucose, or there's no glucose waiting to come into the cell, then all of these will remain phosphorylated, and so it won't block this LAC-Y anymore, and then lactose will come in. Okay? Okay, so I'm going to go over this one more time just as a recap. There is a video on Canvas. I encourage you to watch it, but I'm just going to walk through this again right now, just get big summary of what's happened. So I'm just going to try to draw this. So yeah. All right, so again, this is what the operon setup looks like, right? And so we have LAC-I is the repressor. So this forms the tetramer that can bind to these operating sites. LAC-Z. So LAC-Z, depending on the concentration of lactose, can have two different functions. So in high concentration of lactose, so we have LAC-Z turns this into glucose and galactose. But at low concentrations of lactose, LAC-Z will change this to allolactose. So this right here is our inducer molecule, so this will help turn on the genes. And then finally we have LAC-Y, which imports lactose from the environment. So when will E. coli turn on the lac operon? So when there's no glucose and lactose, but it's positive in lactose, right? So when there's lots of glucose around, we have the PTS blocks, LAC-Y. And then when there's no lactose, LAC-I binds the operator sites, right? And so there's no transcription. But when we have lactose in the cell, so the LAC-Z and the LAC-Y are getting made, but essentially the allolactose will binds LAC-I, and then we get the genes are on. So the LAC-I will release, and you'll get transcription of the genes. Yes?
Does LAC-I repress itself?
Because of this. So I guess it depends. I'm not sure what the rest of the promoter looks like for LAC-I, but again, it's one of those things that if there's enough of it around, that it makes sense that it would repress it, right? Like it doesn't need to have all these extra molecules of LAC-I. Yes?
Do you need cyclic AMP for the mechanism to work, or can you go without cyclic AMP?
It'll go, it's just not going to go as well, right? I guess I'm not sure how often the cell needs lactose and if that's ever going to be considered not starved.
Yeah.
So I'm not sure. Yeah.
What is the thing next to LAC-I binds to? No,
No transcription. TXNS for transcription, yes.
Does LAC-Y work with LAC-Z at low concentrations?
LAC-Y and LAC-Z are independent of each other. Yeah. All right. Okay, so there are many, many, many other operons. Your book talks about the arabinose operon. So I'm not going to go through the mechanisms of this one. Just know that this one's a special case because it can act both as an activator and a repressor. So there are many different functions of these different regulation operons. So the one that we are going to talk about now, so this one has an example of derepression. So this one is the tryptophan operon. So it's also kind of unique because it has both repression, so protein repression, and then attenuation, which is an RNA mechanism. So this one now, instead of controlling the catabolic enzymes, this one's actually controlling biosynthetic pathways, so it needs to make tryptophan. Oops. So for the repression part of the operon, so tryptophan binds to TRPR, so it's a repressor protein, resulting in, so in this case, so tryptophan is your co-repressor, right? So it needs to bind to the TRPR repressor protein for this to then bind to the operating site. So essentially, if you think about it from the cell's perspective, if it has lots of tryptophan, I don't need the enzymes to make it. So tryptophan's going to bind to the repressor, and then it's going to block the genes. And then when there's running out of tryptophan, this will come off, and then back on to make the genes to make more tryptophan. This one has a second control mechanism, and it's to be used for fine-tuning. So this one is a transcriptional attenuation mechanism. So this relies on the fact that transcription and translation are happening at the same time. It uses the ribosome to be able to sense the level of amino acids. So another term for this specific type of attenuation, because there are many other types of attenuation mechanisms, this one is also known as ribosome-mediated transcriptional attenuation. So the DNA region between the operator and so before this first TRPE, the first structural gene, is where the attenuator lies. And so what happens is there's actually also this leader sequence, which refers to a peptide. So there is a start codon, it encodes for this peptide that essentially has no enzymatic function. But the thing that it has is, the key thing that it has is these back-to-back trip codons. It also has a stop codon because it's not supposed to be making anything, so it makes sure that it stops the translation. And then it has 4 complementary nucleotide stretches, so we'll talk about those. So the attenuator is this stretch of RNA, and it has different options for binding. So for the first options so these are the different regions. It has, the first options is for when 1 and 2 bind, and 3 and 4 bind. So if 1 and 2 binds together, that means 3 and 4 will bind together. this results in the formation of an intrinsic terminator hairpin. So remember this from row independent transcription termination. So it's this terminator formation with the U-rich sequence at the end. So if this forms, no transcription, right? The other option is that 2 and 3 will form. And I think there's I put a picture on this on the next slide. But essentially 2 and 3 will form, and the binding of 2 and 3 is actually a little bit more stable. And so if those two form, then I'll just try it. So if you get 2 and 3 forming here, 4 is over here, you have all the U's here, and transcription will keep going, and 1 is out over here. So you never form this hairpin, and so transcription can keep going. So this 2 and 3 formation is called the anti-terminator. This picture here. So the U's, so the U's will be following this region here, this U-rich region here. Okay, so what controls which hairpins form? So when there are high concentrations of tryptophan, the ribosome will translate the leader, but there's a stop codon, and so it'll stop at the stop codon, and essentially it will, when it does that, it sequesters region 2. So now region 2 is tied up, and so then 2 and 3 cannot form, right? So 3 and 4 now are able to form, and so then you stop transcription. So again, high tryptophan, I don't need this genes, so stopping transcription there. In low tryptophan, the ribosome will stall at the double tryptophan codon. So there's strategically placed 2 tryptophan codons next to each other. If there's not enough tryptophan in the cell, the ribosome is just going to be waiting, right? It's just going to sit there waiting for the tRNAs with the tryptophan on them, and it's not going to be able to do anything. So while it's stalling, it's stalling here in this region 1. It's kind of sequestering region 1. So now 2 and 3 can form. And now that leaves, and so now you can get transcription of the genes because it's out of tryptophan. Yes?
Isn't it gonna fall off? Like this,
Which one?
isn't it like a hairpin?
This one, this hairpin here? Is this one you're talking about? So it only falls off if the hairpin's followed immediately by the U's. So at the base of the hairpin, those 2 things are necessary for it to fall off. If it just has a hairpin with no U's following it, it'll just keep going even.
The U's on the RNA.
Yeah. Okay, again, there's a video on Canvas. I will make sure that, so I'm just gonna draw the attenuator again, just to make sure that, because that's the most kind of unique thing about this one. So what happens in this case is that we have, so I'm just gonna draw this out this way. So essentially you have the 2 trip codons here. You have region 1, there's a stop codon here. You have region 2, region 3, region 4, and then there's a bunch of U's right after region 4, right? So if you have high tryptophan, you're going to get region 1, region 1 and 2 are going to bind to this hairpin, and then so that allows 3 and 4 to make this hairpin right here. And so this is your terminator. So we get no transcription. So in the case where there's low tryptophan, so you have the two trip codons here. So the ribosome's going to come and it's going to sit here. But essentially what that's also in here is partially region 1. And so now as soon as, so this is coming and you're imagining that this is happening at the same time as transcription, right? So transcription translations are kind of going along like this, following each other. So I shouldn't have drawn, this is not available yet, but what's going to happen then is as soon as region 3 is transcribed, it's going to bind to region 2. And then eventually it will finish this off and region 4 will be there. So even if the U's are here, even if the RNAP stalls here, without the hairpin formation, it's not going to fall off, and so it'll keep going. Any questions about the tryptophan operon? Yeah.
What's the left molecule for the low?
This is the ribosome. Sorry. Is this clear? Yeah.
So it falls off at the U?
It falls off here, in this case, only in this case. Any more questions? Okay, we're gonna have our next quiz now. So